Scan now. Meet Sovi, your new AI study buddy.
Competitive versus non-competitive sounds tricky, but this one moves fast. Focus on where the inhibitor binds and the correct question basically reveals itself.
Competitive inhibitors directly compete with the substrate for the enzyme's active site, while non-competitive inhibitors bind somewhere else. So the key distinction is location of binding, not product formation.
That means the best question asks whether the inhibitor attaches at the active site or at an allosteric site, because that separates competitive inhibition from non-competitive inhibition cleanly. Also, increasing substrate can overcome competitive inhibition, but not non-competitive inhibition, which again points back to binding site as the deciding idea here. Well done. Option D, scan now. Avoid rushing past key signs. Trust the given details and check each step before locking your answer.